Guys, you are watching Biology with Insure. So for today's video we are going to be discussing about proteins, which is a section of the second chapter, biological molecules, of your AS biology. The proteins is a huge part of this chapter, and it may take about 2 to 3 videos to complete proteins, so just bear with me and I'll do my very best to ensure that it is completed. Okay, and one more thing, I just wanted to say that I'm really sorry for the fact that a video was uploaded after a long time. So there are 2 reasons for that. The first one being that I was a bit busy with some things, and number 2, we were having some issues, just technical issues. When I wanted to upload a video, we were having some issues, so I apologize for that. And for to make up for that, I'll be uploading 2 or 3 videos in 1 week for you guys, right? So stay tuned and keep sharing this around. Now without further ado, let's get started. Okay, so proteins. This is just basically your O levels or GCSEs, nothing new. Okay, right. So proteins. One of the main important things to understand is that proteins are made up of carbon, hydrogen, and oxygen. Now the 3 common elements that are also found in your carbohydrates and lipids, but then there's this additional element here that is found in your proteins, which includes nitrogen. So nitrogen is always present in proteins, right, along with carbon, hydrogen, and oxygen. And understand this carefully when I say this, and maybe sulfur. Now carbon, hydrogen, oxygen, and nitrogen are always present in your protein, but sulfur may or may not be present. It depends on the type of protein, right? And note that the sulfur is attached with the R group. Now what is an R group? We'll talk about this later, okay? So I'll explain that in detail. An amino acid, which is actually the simplest form that makes up a protein molecule, as we all know. So what happens, we're going to talk about how protein is made, right? So the simplest form, your several amino acids first join together. So they join together to form this long chain, which is called a polypeptide. So let me just demonstrate this through visuals for your understanding. Let's suppose that this is one amino acid, another second amino acid, let's say a third amino acid, and they can be the same, they can be the different. So let's use another colored pen to show maybe this is another type of amino acid. It can be the same or different, okay? Now, so what happens is that they're going to join together, right? So they're joined together. There's going to be a bond between each amino acid, a bond between this and this amino acid, and a bond between this and this amino acid. If you're following my cursor, you'll understand it. So this bond is actually referred to as a peptide bond, right? So remember that it's a peptide bond, and this long chain here that you can see, it can be longer, by the way, but this is just for your understanding. This long chain here is called a polypeptide. Now what does poly mean? Poly means many. Peptide, many peptide bonds. 1, 2, 3, 4, many peptide bonds are here. Okay, so this is a chain now, as you can see. So this is how a polypeptide chain is made. Polypeptide chains and a protein are different. So polypeptide chains make up a protein, as a number of these chains, not just one, like several of them, wrap around each other, right, to form a complex 3D molecule called a protein molecule, right? Let me demonstrate. So for example, this is one polypeptide chain. Let's say this is another polypeptide chain. They can be different or same, it depends upon the type. Another polypeptide chain. So basically you can see they're wrapped around each other in this complex way, and it has a 3-dimensional structure, which is nothing but a protein. And this here is how a protein is made, right? Just to recap, you've done this in your O levels and IGCSEs, but then again, a recap is always good. There are about 20 different types of amino acids, and the different combinations of these gives rise to the different types of proteins that exist today. Like the protein in our nails is very different from the proteins in our hair, from our skin. I'm just giving an example. Like there are different types in chemical composition. They can be similar. Like for example, the protein in your hair and nails are a bit similar, but a little difference in the composition, right? But what I'm trying to say is that 20 different amino acids means many different types of proteins, and that's how like the words in English we have. Like for example, there are many letters, and their different combination gives rise to the different types of words that exist. Okay, so this is a diagram here that you have in front of you that's explaining how protein is made, right? So just gonna explain it real quick because we just discussed it. These are individual amino acids, and this is a polypeptide chain that's been formed from these amino acids joining together. And you can see that this is a peptide bond, which I have labeled here, that is linking these amino acids together. Note that their color is representing their type. So notice there are two browns here, which means there can be same amino acids or maybe even different. Okay, and then how does this convert into a protein? A polypeptide chain converts into a protein when these chains wrap around each other, just like I discussed earlier. Notice how this is wrapped, coiled, like you can say wrapping, coiling, condensing. These are some of the terminologies you may have to use in your P2. So this is how a protein is made. Now earlier I said that many chains wrap around each other. At times just one chain can wrap around on its own to give rise to a protein. So this is also possible. Now this is a bit additional extra stuff, not extra, it's part of the syllabus, but new for you in AS biology. So till here you studied this till your O levels in IGCSEs, but now a protein has its types as well, and this is going to be extremely new for you in AS biology. For instance, protein has a primary structure, has a secondary structure, tertiary structure, and quaternary structure. So basically when a protein is made, from step number 1 and step number 2, a protein is then converted via step number 3 into its primary structure, and then a primary structure is converted into a secondary structure, and then a secondary structure is converted into a tertiary structure, which is lastly converted into a quaternary structure. So these are some of the things you need to know, and we're going to discuss primary, secondary, tertiary, and quaternary one by one with some examples, their diagrams, how they look, how they're made, and it's going to be very easy, but it's a bit lengthy. That's only the problem, but the rest I'm going to do my utmost to ensure that it is completed, right? Then we're going to understand some functions of proteins, just a recap of what you've done in your O levels and A levels. For instance, forms enzymes, hormones, antibodies, and hemoglobin, which is present in your red blood cells. The cell membrane is made up of different proteins and lipids. The reason why I'm saying proteins is because proteins make up your cell membrane, and our main focus is proteins. Responsible for the clotting factor, your platelets are made up of proteins, and your fibrinogen, prothrombin, they're all protein, okay? So we're not getting there, it's something that we've studied, but just understand it. Now let's understand something new that is in AS, something new that you've never encountered before, amino acid structure. Okay, amino acid, as we discussed, is the simplest form of protein, but we're going to be discussing its structure, chemical structure. Okay, we know the carbon has 4 bonds, right? That's basic chemistry, and this is actually a permanent structure of an amino acid. Okay, so you can see that the central carbon has 1 hydrogen atom, the first bond. The second bond has an amine group, or you can call it as an amino group, which is nothing but your NH2. And then the third bond is your carboxylic group with carboxylic acid, which is your COOH. And note that in your carboxylic group, there's always a double bond between one oxygen and carbon, and then the OH is on the other side. This is a functional group, okay? And then this R that I was talking about, in which we were discussing that if sulfur is present in your protein, it's usually in the R group, which is this, okay? The R group basically means this can be anything. This can be maybe, let's say, calcium. This can be, let's say, sodium hydroxide. It can be literally anything, depends upon the type of amino acid. This is what helps us to identify the type of amino acid. What I'm trying to say is that the amine group, the hydrogen, and the carboxylic group are always there in every amino acid. These 3 are fixed. The only differentiating factor is your side chain / the R group, as this helps us to identify the type of amino acid. It can be the same. If it is the same, then the type of amino acid is the same. So this basically is a short way of writing. Maybe let's say glycine, which is a type of amino acid, does contain an amine group, a hydrogen, and carboxylic acid. And for its R group, it may contain, let's see, it may contain maybe potassium. That can be anything, right? And since it contains potassium, it allows us to identify that this here is glycine. That's why this is unique, okay? So in every amino acid, only the R group differs, just like I discussed, and this allows us to identify the type of amino acid. Yeah, so all amino acids contain an amine group, NH2, carboxylic group, COOH, and a hydrogen. As these are fixed, remember only the R group changes. Then this is something important. Amino acids link up together, just we discussed, to make polypeptide chains in a condensation reaction. When I say condensation reaction, what I'm trying to say is that we've actually discussed this in our previous videos, but I'm just going to give a recap what a condensation reaction is. In a condensation reaction, two simple molecules, or maybe three, combine together, and when they combine together, they form a complex molecule. And in this process, a bond is made, not broken, it's made. And during the formation of this bond, or several bonds, a water molecule is eliminated. It is removed. So at the end, you have your product plus water molecule. And this is exactly how your amino acids link up together to form a polypeptide. And that is why a water molecule is also made, to form a polypeptide and a water molecule, right? And hydrolysis, which is another type of reaction, is the reverse of condensation, and water is added to break something complex down into simple. And in here, the water is added instead, and with the product, no water is made, right? So just try to remember this, because you know they're gonna be asked in the exam, and they're quite important. Okay, now let's talk about the condensation reaction, which is right here. Okay, before I do that, let me just quickly rub this off. Sorry for that. Okay, so we're talking about how a condensation reaction occurs between two amino acids. So first, let me explain it. Now this here is one amino acid, and this here is, yeah, this is one amino acid. This is your second amino acid, right here. Okay, so you can see here. I'm just gonna give a highlighter that this is your central carbon, your amine group, and your carboxylic acid, and your hydrogen, and the R group right here. And it's the same here, carboxylic acid, amine group, hydrogen right here. Okay, now that it's been understood, now I'm going to talk about how water is removed, right? Water is removed in a condensation reaction, and that's why water is made along with the product, and a bond is being made. So the OH from, one second, yeah, so this OH here from the carboxylic acid of the first amino acid is removed along with the H from the amine group. I'm circling the amine group, by the way. Oops, one second. Let me explain that again. I confused it a little bit. Okay, so here's the thing. OH, as you can see, is right here from your carboxylic group, which is right here from the first amino acid, is removed along with the H right over here from your amine group of your second amino acid. So OH here and H here means H2O. So they're removed together, which means the water is being eliminated, and a bond is made between this carbon and this nitrogen, right? Yeah, it's circled here, the red here. This carbon here of your carboxylic group, and nitrogen here of your amine group. And this bond between this carbon and nitrogen is called your peptide bond, and this is the polypeptide. And don't forget to show plus water molecule. You may get to draw this for like 3 marks, so you have to show it exactly like this to get 4 marks. Like write down that this is a condensation reaction, then do show that water is removed like this by showing negative H2O, and then you have to label the bond here as peptide bond, and then you have to show plus H2O. Do it this way and you'll get 3 out of 3 for that particular question. Okay, so let's understand it again because it's written down here. So in the formation of a peptide, which is the same as a polypeptide, that is, when 2 amino acids link together via a peptide bond in a condensation reaction. So the OH from the this is fixed, by the way. OH is always from the carboxylic group of the first amino acid, and H is from the amine group of the second amino acid, are both removed together, which is basically the removal of H2O, forming a peptide bond between the carbon of the carboxylic group of the first amino acid and nitrogen, which is right here, of the amine group of the second amino acid. And this is where a peptide bond is made and water is formed. Note that if you add water to this, okay, this is actually showing the formation, If you add water here, you would actually be breaking this bond to form this again. So condensation and hydrolysis are both opposite. So we understand here that a protein can be broken down into its amino acid, into its constituents via hydrolysis, which is the addition of water as the water is needed to is added to the polypeptide chain, and hydrolysis means bond breaking. So you're breaking the peptide bond. This is the complete reverse of the condensation reaction shown behind. Proteins are broken down like this via hydrolysis in the stomach and small intestine, so the amino acids can be absorbed by the body in their simplest form. This reaction here, which is the complete opposite of what we just studied, actually occurs in our bodies during digestion, right? This is basically your polypeptide. As you can see, we have labeled the peptide bond. I just rubbed this, used it for my previously to teach someone. Okay, you can see here, this is one amino acid and the next, and this is the peptide bond right here between the carbon and nitrogen. Dipeptide means two amino acids combined together. It's simple. So water is added here. As you can see, water is added. You need to show it like this, by the way, to break this bond, and this means it's being broken down, okay? And when it's broken down, you can see that two separate amino acids happen. This is one amino acid and this is the other amino acid. Notice it's the complete opposite, as OH, if I just circle it for you, is added to the carboxylic group of the first amino acid, and your H is added to the amine group of the second carboxylic acid, resulting in two separate amino acids. The complete opposite of what you just studied. Okay. So in hydrolysis of a protein molecule, the peptide bond between the amino acids is broken down, leading to further reaction. And when the peptide bond is broken, a water molecule has to be added in order to break the bond. So to break this, you need to add water. Okay, from a water molecule. Okay, so remember that OH that came here and the H that came here, they all come from water, the water that you're adding. So OH is added to the carbon from the carboxylic group of the first amino acid, and H, which is right here, is added to the nitrogen, which is right here, of the amine group. This whole thing in blue is the amine group of the second amino acid, and the H added to the carbon of the carboxylic group of the first amino acid. Okay, so that's let's keep it till here for today. I'm sure this is quite a lot to take in, so just try to understand this, try to draw the diagrams, like this diagram over here, and this is like the most important diagram of all, this one. You get this for sure in your paper 2 for AS biology for like 3 to 4 marks, mostly 3 marks, right? So just try to understand this all, and then in the next video we're going to be discussing about primary, secondary, and tertiary and quaternary structure. So that may take about 2 or more okay, 2 videos is fine. Yeah, I think 2 should be fine. It's 2 videos or so, because I want to complete each and everything. So I hope you enjoyed this video. I hope this helps most of you guys out, and if it did, don't forget to like my video and subscribe to my YouTube channel. And if you're watching this on Facebook, like my video, react to it, and follow it on my Facebook page. Give it a like. The more the followers, the better, because I am making free quality content for you guys. So make sure to hit the like button, subscribe, comment, do all the things that you can to support me, and I'll support you back by making free, awesome, and quality content. So that's it for today. See you this week. As I said, I'll be making more content for you guys to make up for the loss. Keep learning. Have a great day. See you this week.